Hi everyone, welcome back to my A-level biology lecture series. In this lesson, we will explore chromatography. At the end of this lesson, you will be able to describe and use chromatography to separate and identify chloroplast pigments, and the reference should be made to the RF value, or known as retardation factor, in identifications of chloroplast pigments. All right, let's get started. Chromatography is a technique used to separate and identify the components of a mixture. In this diagram, you can see that we can use chromatography to separate different pigment or different solutes by using chromatography. You can see that the red color pigment, blue, the green can be separated, into the green and yellow pigment. Initially, they are in a mixture form. So this separation occurs due to the difference in how each component or the solute move through a stationary phase under the influence of a mobile phase. We are going to discuss about stationary phase as well as the mobile phase. So in the analysis of chloroplast pigments, chromatography is used to separate chlorophyll A, chlorophyll B, xanthophyll, as well as carotenes. So for the separation of chloroplast pigment by using thin layer chromatography, we need a thin layer chromatography sheet or a strip, which is shown in this diagram. So this chromatography strip consists of a base, which acts as a solid support layer that provides structural backing. And on top of this base, we have this stationary phase, which is a thin polar layer absorbance material, which can be a silica gel, which spreads over the base. So this phase interacts with the substances by the process known as adsorption, not absorption. Adsorption basically means interaction. So in chloroplast pigment separation, TLC separates pigments based on these two factors. First, the pigment solubility in the mobile phase, the solvent that dissolve the pigment, as well as their adsorptions to the stationary phase, which is silica gel on the TLC. So again, the adsorption can be interpreted as the interaction between the pigment and the silica gel. So what is meant by solubility in the mobile phase? So the pigments that are more soluble in the mobile phase will travel further up the TLC plate or TLC strip, and therefore they also move faster with the solvent front. Imagine that if the pigments solubilize well in a particular solvent, they tend to be carried by this solvent and move further from the origin line. And this is because they spend more time dissolved in the moving solvent than attached to the stationary phase, which is silica gel. How about adsorptions to the stationary phase? So the stationary phase, they are polar, and they interact with the molecule through the interactions known as adsorption. Polar pigments are more strongly absorbed onto the stationary phase, and therefore they move slower and they stay closer to the origin line. You can try to imagine that when you go to a mall, and then you go into every shop, you interact with every shop, shop A, shop B, shop C, and so on. So therefore, you spend more time to reach your destination. So same goes to the pigment. If the pigments tend to interact well with the stationary phase, the silica gels, so therefore they move slower and they stay closer to the origin. So in contrast, non-polar pigments absorb less, and travel further. So the distance a pigment travel on the TLC strip is determined by the combination effect of solubility in the mobile phase, means the solvent that carry the pigment, as well as the adsorptions to the stationary phase, the silica gel. So this interplay between solubility and adsorptions result in each pigment having a characteristic position on the chromatogram. So what is a chromatogram? If you look at this diagram, this is a chromatogram. And you can see that at the end of the process, we have the origin line. Okay, this is origin line. And this is the solvent front. So basically, when solvent travel up to this level, so the pigments that dissolve by the solvent will move together. So based on the solubility and the adsorptions of the pigment, they can be separated. Okay, so look at this pigment A. Pigment A move further away from the origin, the origin line, and the pigment A is far away from the origin line. So this pigment A definitely has high solubility in the mobile phase and low adsorptions to the stationary phase. Let's look at this pigment B, which is closer to the origin line. So therefore, pigment B has low solubility in the mobile phase and high adsorption to the stationary phase. So the interplay between the solubility and adsorption and finally determine the positions of a pigment on a chromatography strip or chromatogram. So what are the procedure for the thin layer chromatography to separate chloroplast pigment? First of all, we definitely need to extract pigment. To extract pigment, we can actually obtain 20, for example, 50 grams of fresh leaf, and we place it in a mortar. By using a pestle, okay, we can grind this fresh leaf, okay, in acetone. So acetone is a solvent to dissolve or to extract the so to absorb the chloroplast pigment. This is very important to use acetone because acetone can dissolve both polar and non-polar chloroplast pigments. So by grinding this leaf, we will be able to release chloroplast pigment, and this chloroplast pigment is the chloroplast pigment dissolved in acetone. So we will continue to grind until a dark green solution is formed, and we must filter the extract through the muslin cloth to remove the plant debris. Okay, so we can collect this pigment extract, means the filtrate, in a clean beaker. To concentrate this pigment, we can always place this beaker of pigment extract in a fume hood to evaporate excess acetone. At the end, we will be able to get a concentrated pigment. So we are done for pigment extractions. The next part, we need to prepare the TLC strip. So very simple, we use a pencil, generally draw an origin line about 10 millimeter from the bottom of the TLC strip. So from the picture here, diagram here, you can see that the pencil line indicates the origin line. And then by using a fine brush or capillary tube or even the pinhead, we can transfer the pigment extract onto this origin line. So we allow it to be air dry and we reapply, okay, few more times to get a concentrated spot. Be careful, we must ensure that the pigment remain concentrated. At the same time, it does not spread too widely. So every time we allow the strip to dry and to concentrate the spot until we get a very dark spot on the origin line, and we allow the strip to dry completely before we continue to the next step. So we have to separate and to develop the chromatogram. So we can pour a small volume of suitable chromatography solvent. So in the lab, normally we use a cyclohexane with ethyl ethanoate, okay, a mixture of these two solvents into a boiling tube. But this is the trick. We must ensure that the level of this chromatography solvent is below the origin line. And carefully place this TLC strip vertically in the solvent and cover the tube with the rubber band to minimize the evaporation. And bear in mind that chromatography solvents are volatile, and most of these organic solvents are toxic. So we allow the solvent to rise by capillary reactions, okay, as shown in the diagram. So you can see that when solvent move up, okay, by capillary reaction, so you can see that this is the solvent front. So when the solvent move up, okay, by capillary reaction, it dissolve the pigment and carry the pigments together. So the separations of pigment based on the solubility and the absorptions of the pigment. So when the solvent front near to the top edge, about 10 millimeter from the edge, we remove the strip and make sure that you mark the solvent front. This is very important because as we know that chromatography solvent is very volatile. A lot of times when students get excited, when they remove this strip out from this boiling tube, they are so excited and see how many pigments they can obtain, how many pigment they can separate and so on. At the end, they realize that there's no more solvent front. You can't see solvent front anymore when the solvent is totally evaporated. So first thing we have to do after we remove the strip, draw a solvent front. After that, we can analyze the pigment. So let the TLC strip air dry as usual, and observe and records the distinct pigment bands. So to identify what are the pigment available in the sample, we need to calculate retardation factor or retention factor. In short, we call it RF value. So retardation factor is a ratio used to quantify how far a substance travels during chromatography relative to the solvent front. So the formula is distance moved by the pigment divided by the distance moved by solvent front. So the RF value is a unitless value, no unit, ranges from 0 to 1. So each pigment has a characteristic RF value depending on the condition like solvent type as well as the stationary phase. To calculate RF value, first we need to measure the distance from the origin to the center of the pigment band. And we need to measure the distance from the origin to the solvent front. And we can calculate the RF value by using the formula distance moved by pigment divided by the distance moved by solvent front. Let us calculate the RF value and try to identify some of these pigments. So experimental RF value can be compared to the reference value to identify unknown pigment. For example, we have a series of RF value here, include xanthophyll, stigmasterol, pheophytine, and so on. Now, this is a chromatogram. So first of all, this origin line, for example, and we have the solvent front. We can always measure the distance moved by the solvent front. So in this case, 95.5 millimeter. So for pigment A, we can measure the distance from the origin to the center of the pigment. Let's say 90.5 millimeter. And by calculating RF value, we get 0.95. Refer back to the reference value. We know that pigment A is beta carotene. We can do the same for B, pigment B. So we measure the distance, 42.0 millimeter, and we calculate the RF value, we get 0.44. So based on the reference value, we know that pigment B is chlorophyll A. And for the pigment C, we measure the distance, 30.5 millimeter from the origin, and the calculated RF value is 0.32. So based on the reference value, again, 0.32 actually is chlorophyll B, and we can do the same for pigment D. Okay, so the distance is 15.5 millimeter with the RF value 0.16. From the reference RF value, it is xanthophyll. Okay. However, you can see that some of these RF value, they are very, very close. For example, chlorophyll A with 0.44 and alpha tocopherol with 0.45. So we might need to further confirm, okay, the identity of the pigment. The question is how? So to confirm the pigment identity, we can use a method known as co-chromatography. So in this method, we will be using a pure standard, a pure pigment in this case. So we can spot the pure pigment. For example, we want to know that is it confirmed? Can we confirm it is chlorophyll A? We can always get a pure chlorophyll A, and then we use a TLC TLC strips or TLC sheets. Then we can have one spot for the pigment extract, the sample, and another spot for the pure pigment. So we call this method as co-chromatography. So we develop the chromatogram in the same solvent system under identical condition. So what are the confirmation criteria? So to confirm, first, the pure standard and the unknown must produce a single overlapping band, and they are likely be the same compound. And both must have the same RF value and with similar color. So why similar color? Because it's totally based on the concentration. So in the pigment extract, the concentration might be low. So the pigment color, after we have done the separation, might be slightly paler compared to the pure pigment. So by doing so, the co-chromatography controls for the experimental variation, for example, the temperature, as well as the solvent concentration, type of solvent, because both of them use the same solvent, use the same stationary phase, same TLC sheet. And with that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any question, feel free to drop them in the comment section. See you in the next lesson. Have a great day.